oral cavity, then esophagus, then stomach. Today we'll discuss, we'll start from small intestine. So from small intestine, we know that the main function is absorption, and we know where there is absorption, there will be simple columnar type of epithelium, right?
Sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So in small intestine, we know that everywhere in small intestine, if we start with mucous membrane, submucous layer, muscle layer, and serous layer. So mucous membrane, there will be
Have a look at these pictures.
simple columnar type of epithelium. You know that in simple columnar epithelium, there is the cells are elongated, the nuclei are also elongated, and they rest on a single basement membrane. Now, the specialty in the small intestine is that the epithelium and the lamina propria here, they actually show some finger-like projection of mucosa. Okay. So these lining epithelium, they become like this. The cell and as well as with the lining epithelium. Okay. So these finger-like projection, they will be called villi. If you see the figure here, so they are the epithelial cell. You see here, these are the cell, these are the lining epithelium, and these finger-like projection of epithelium with the lamina propria, they are called villi. So if we see this structure, so they are villi. Okay. So, and we know that there is another term that is called microvilli. So now, what is the difference between villi and microvilli? Can anyone tell me?
স্যার মাইক্রোভিলাই হচ্ছে এপিথেলিয়াল সেলের যে প্রজেকশন গুলো ওগুলো।
Yes, if you see here,
Sir, villi হচ্ছে mucosal finger-like projection of mucosal fold, and যে enterocyte থাকে villi এর মধ্যে, সেইখানে যে স্যার finger-like projection থাকে সেটা হচ্ছে microvilli.
okay. So microvilli is a feature of cell, individual enterocyte or simple columnar epithelium. They are a fold of the lining epithelium, sorry, folding of the cell membrane, but the villi are the folding of lining epithelium with the lamina propria. And in the lining epithelium of the small intestine, we can see some intestinal glands. They are called crypts of Liverpool. If you see here, intestinal gland here, these epithelial cell invaginates to form some glandular structure. They are called intestinal gland or crypts of what? We have completed all the class. Crypts of Liverpool. Spelling, always check the spelling. Then between the villi, these intestinal glands, they will open in these crypts. Okay, so these are the intestinal glands of crypt of liver cone, and they open here. They are called the crypts of the intestinal gland. Okay. Now, in the crypts of liver cone, there are different types of cell. First of all, there will be some undifferentiated cell. Undifferentiated cell means they do not have any specific structure or specific function. They are undifferentiated, and there will be some absorptive cell. They will absorb the digestive end products. There will be some goblet cell, and there will be one important cell, that is Penneth cell. So Penneth cell is one important cell of the intestinal gland, and their main function is to provide immunity. So in the crypts of liver cone, cells in crypts of liver cone, there will be undifferentiated cell, absorptive cell, few. There will be some goblet cell, Penneth cell, and some enteroendocrine cell. Now, this is all about the mucosa. Now, submucosa. Okay. So, in the small intestine, there are three parts. First of all, the proximal part, that is called duodenum. Then the second part, that is called
jejunum, sir.
jejunum, and last is ileum.
Ileum.
So, in this submucosa, we have the slide of the duodenum in your college, and the most characteristic features of the duodenum is there will be some gland present in the submucous layer of the duodenum, that they are called
Brunner's gland.
Whenever you will get the slide of duodenum, you will be asked, how will you identify this slide as duodenum? You will say that presence of gland in the submucous layer. Then the second question will be, what is the name of the gland? So the answer will be Brunner's gland. Now, in the ileum, there will be no such gland or Brunner's gland. In the ileum, it is closer to the large intestine. So we know that the distal, the more distally you go in the intestinal tract, there will be lymphatic tissue will be more. So in the ileum, the submucous layer will contain some gut associated or mucosa associated lymphoid tissue, and there will be also some Peyer's patches. You know what is Peyer's patches? They are
aggregation of lymphoid
lymphatic
tissue.
nodule.
Yes.
Then muscle layer, you know, it is common for almost all the that part, outer longitudinal and inner circular. And serosa is visceral peritoneum. Now, if you see the figure, we have covered this figure. Now, this is a structure of intestinal villi. It is very, very important both for written and for OSPI. Now, a villi is actually the fold of lining epithelium as well as with the lamina propria. So the contents of the lamina propria, they will be present in the lumen, sorry, in the villi. If you see this figure, if we divide this figure into three parts, this is here. So the villi will contain vascular structure, that is artery and vein, and the capillaries, as well as we know that the intestinal mucosa are their function is to absorb the digestive end product. There are some smaller end product, for example, glucose or the carbohydrate end product, then some amino acid or fatty acid that are fat and protein digestive end product. And there are some larger molecules, for example, chylomicron or something like that, and they will be passed through the lymphatic channel. So the villi will also contain some lymphatic capillary, and they are called lacteal. And to maintain the shape of the villi, there will be some smooth muscle. So the core of the villi will contain vascular structure, and some lymphatic structure and some smooth muscle. Clear everyone about the structure of the villi?
Yes, sir.
Yes, sir.
जी sir.
Then if we see the histological structure, so this is the histological structure of duodenum. So how will you identify duodenum? The lining epithelium is simple columnar, and there will be mucous fold there, villi, and the presence of duodenal gland in the
কলকাতা মতো যাবো, ইন্ডিয়া যাবো, পরে ব্যাংকক যাওয়ার ইচ্ছা আছে, আস্তে আস্তে একদিন যাওয়া যাওয়া করবো ইনশাল্লাহ। লস করার তো আমার
কিছু বলবেন, কোনো সমস্যা? হ্যালো, কেউ কিছু বলছিলেন? কি ব্যাপার? যাই হোক, so in the duodenum there will be villi and there will be presence of duodenal gland in the submucosa. These are the mucosal gland or Bunner's gland, and this Bunner's gland is very clear in the slide that are in your college. So this is mucus layer, submucous layer, these two muscle layer, and the serous layer is not present here. This is another figure on the histological slide figure হচ্ছে আপনার duodenum. So here these are the villi. Okay, these are the submucosal Bunner's gland. This Bunner's gland is very important for the identification of duodenum. Now the later part, sorry, before going to the later part, so this is the slide you will get in your college. So if we get this slide in the exam or prof, you will say this is a histological. This slide is of the slide of duodenum because there are numerous intestinal fold, there are villi, presence of submucosal gland or Bunner's gland. The lining epithelium, if you focus zoom here, the lining epithelium are simple columnar epithelium. Okay, so lining epithelium, then features of the submucous layer, So these two are the main identification point of the duodenum. Now in the later part, in the jejunum, in the submucous layer, there are no Brunner's gland, but in the submucous layer there will be some lymphatic nodule. The lining epithelium are same, simple columnar epithelium. There will be presence of intestinal gland. And in the jejunum, there is an important cell that is called Pennant cell. So if anyone asks where you will find Pennant cell, that it is important for the MCQ, the answer will be jejunum. Can you hear me?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Okay. And this is the physical structure of jejunum, ileum. So the more this we go towards the distal part of the intestine, the number of lymphatic nodule or the lymphatic tissue will be much more. So it is a structure of the ileum, and you can see here in the submucous layer, the number of Pierce patches or aggregated lymphatic nodules are much more. So in the submucous layer of the ileum, there will be no Brunner's gland. There will be numbers of Pierce patches. And the features of the lining epithelium same. There will be intestinal gland in the lamina propria. There will be villi. কার কথা শুনতে পাচ্ছি মাইক্রোফোনে? কার মাইক্রোফোন অন করা বলুন তো? মাহফুজুর রহমান রায়হান, আপনি আছেন এখানে? মাহফুজুর রহমান রায়হান। মাহফুজুর রহমান রায়হান, আপনি কি শুনতে পাচ্ছেন? I think he is busy somewhere else. Okay, now the large intestine. Any question in the small intestine part?
Sir, Pennant cell की ileum में पाओ जाए ना?
Not that much. It is the characteristic features of the jejunum. And in the duodenum, you will find the Pennant cell. Sorry.
Brunner's gland.
Sorry. Now, large intestine. So again, we will discuss like this: mucus, submucous layer, muscle layer, and serous layer. So in the large intestine, we will not find any villi. So no villi. That's the main features. Then the epithelium lining is simple columnar with microvilli, same. But the intestinal gland in the large intestine, there will be the number of the goblet cell will be much more. Okay, so the more distal you go to towards the digestive tract, the number of goblet cell will be increasing, and the number of lymphoid tissue will be increasing. Okay. So in the small intestine, the intestinal glands or the glands strips of Liverpool, there were few numbers of goblet cell, but if you see here, these whitish cell, this white structure, all these things are goblet cell, because we have learned that the function of the goblet cell is to secrete
Mucus.
what? Mucus. And in the traditional staining, the mucus doesn't take any stain, so they appears whitish in the histological slide. So in the slide of large intestine, the number of goblet cells will be much more, and we will identify them as they will not take any stain, so there will be this white gap here. So there is no villi. This depression here, they are actually the crypts of Leprechaun or the intestinal gland. If you see the histological structure here, you can see they are the intestinal gland, and they are the all this cell. They are the goblet cell, and in the submucous layer there will be lymphatic nodule. So all these are the histological slides of the large intestine. You can see here the main identifying features of the large intestine is presence of numerous goblet cells in the mucous layer and presence of lymphatic nodule in the submucous layer. This is the slide of large intestine that is in your college. So there is no, it is not that much clear, but whenever you get this slide, if you focus in higher magnification, you will see the lining epithelium is simple columnar, and in the submucous layer there is no gland. Okay. But the lymphatic nodule or the goblet cell that are not that much prominent, but whenever we will get chance in college, we will try to see the overall full slide so that we can easily understand. Now, before going to this slide, you see here. So serous muscle layer is similar. Then serous layer similar, and submucous layer we have mentioned earlier that there will be no glands, but there will be numerous lymphatic nodule will be present. Now another part of the large intestine, but it is very important. It is appendix. What is appendix?
A tube-like projection from cecum.
So histologically, they are almost similar to that of the large intestine, but smaller in size, and the lumen is also smaller. So lining epithelium, mucosa, submucosa, muscle layer, and serous layer. So mucosa same, simple columnar epithelium. Submucosa is fully covered by lymphoid nodule. Okay, numerous lymphoid nodule will be present in the appendix, and it is the most characteristic feature of the appendix. Then muscle layer same, serous layer same. So this is the histological slide of the appendix. You will identify simply because as appendix is very small, so you will be able to see the whole appendix in lower magnification. That is the first one. Then this larger lymphatic nodule will be clearly visible in the submucosa. So whenever you get a small luminal structure with lots of this nodular structure, that will be appendix. If you see another term here, so these are the mucous layer, lining epithelium same, The larger part, these are the submucous layer. This is one. These are the numerous lymphatic nodule. Hello, শুনতে পাচ্ছেন?
Yes, sir.
Yes, sir.
Okay, this is another diagram of the histological slide of the appendix. These are the lining epithelium. These are the lymphatic nodule. This is the slide that you will find in your college. So all these things are the lymphatic nodule. Whenever you will, it is the most simple slide, very easy slide for prof. Whenever you get this slide, it's simply this, by seeing this lymphatic nodule, you'll be able to identify the slide as a appendix. Now, before this is all about the histological structure, detail of the intestinal or digestive tract. Now, before going to the next part, we'll have to discuss about one thing, that there are different type of lining epithelium present in the digestive tract. If we name them, first one is protective epithelium. Which one is the protective epithelium among these three? One is protective epithelium, one is secretory epithelium, another one is
Lining
absorptive.
epithelium, protective.
Which lining epithelium?
Simple columnar.
Simple columnar epithelium.
Protective is the non-keratinized stratified squamous epithelium. This one.
Non-keratinized.
They will be line. They will line the oral cavity, pharynx, esophagus, anal canal. Okay. That may be sometimes stratified in some places, and sometimes sometimes that may be keratinized, or sometimes that may be non-keratinized. But these stratified squamous epithelium, they are the protective epithelium. Then secretory epithelium, that is actually present only in the stomach. This is not present. The slide is not here because the stomach lining epithelium, they only secrete the gastric juice, but the other epithelium, they can be classified as absorptive epithelium because they are made to absorb the digestive end product. Okay. So protective, secretory, and absorptive, these are the gross subcategories of the type of epithelium that lines the intestinal tract. Now, okay, 55 এখন আসছে। Now, liver. So, along with the digestive tract, there are some accessory glands associated with the digestive tract. They helps in the digestion. The most important of them is liver. I think you have completed the liver items. I am just directly going to the histological part. So histologically, one glandular structure can be categorized into two parts. One is parenchyma and stroma. Now, can anyone tell me what is parenchyma and what is stroma?
So the functional unit of an organ is the parenchyma, and the rest, like the
Functional
connective
unit of
tissue.
an organ, it is actually not a nice answer. So parenchyma, or in case of gland, the secretory part of the gland, they are called the parenchyma, and their fibrous structure or supporting structure, they are called stroma. So in case of liver, the stroma is only 20% and the parenchyma is almost 80%. And these parenchyma in the liver, they are organized into different types of hepatic lobules. And these hepatic lobules are formed by, there are some central vein, and there will be radiating hepatocyte, and they will be arranged in different lobule. We will discuss that now. So if you see the figure here on histological slide, this at first this is the schematic diagram. So what are the types of lobule present in the liver? Anyone?
So classical hepatic lobule, portal
Classical
lobule,
hepatic
and
lobule,
the liver
portal lobule.
acids.
Total
So
liver and liver.
classical hepatic lobule, how they are formed? They are formed by centrally there will be a
Central vein.
Vein.
central
Hepatic
vein, right?
Yes, sir. And
And
on hexa.
on
Hex.
6th angle there will be
Portal
total triads. 1, 2, 3, 4, 5, 6. So this hexagonal shape structure is called hepatic lobule. So how portal triad is formed? By the branch of
hepatic artery, portal vein, and bile ductus.
Arterial, portal venule, bile duct.
Now tell me, what are the function of these hepatic lobule? This is
The endocrine function of the liver.
only endocrine function.
Both endocrine and exocrine.
The nutrient supply.
আচ্ছা আমি বলছি আমি আচ্ছা আমরা একটু পরে আসি first we discuss this. So these around the central vein, this is the central vein, and these are the radiating hepatocyte, and all these hepatocyte, they are encircled by these green dots. They are the bile canaliculi. So if we draw a hepatocyte here, for example, this is a hepatocyte. So bile canaliculi, they will encircle all the part, okay? So they will totally cover the 6th side of the hepatocyte. So these different types of triad lobule, their function is to হ্যালো,
Yes, sir.
শুনতে পাচ্ছেন?
Yes, sir.
Yes, sir, এখন শুনতে পাচ্ছি।
ওকে, সরি, ডিসকানেক্ট হয়ে গেছিল একটু। So now, this is another schematic diagram or histological structure of the hepatic lobule. So in here, these are the this is the central vein. These are the 1, 2, 3, 4, 5, 6 portal triad. So this hexagonal structure, that this is another hepatic lobule. So in your college or the histological slide that you will get in the exam, they looks like this. So you will not get the exact 6 portal triad on the 6th angle, or it is not always possible to see the textbook type hexagonal hepatic lobule. So the main finding is that you will get some this structure, that is the central vein, and you will get around the central vein there will be radiating cells. These are the radiating hepatocyte, and on some corner you will get the portal triad. So the hexagonal shape, it will not always be visible. So this is another schematic diagram and very, very important diagram both for your written, oral, and histological slide related paper. So there are 3 types of hepatic lobule. First one is classical hepatic lobule, then portal lobule, and hepatic acinus. So hepatic lobule, that is the classical hepatic lobule, and we have discussed about this one. So central vein and the 6 portal triad. So this hexagonal shape structure, their function is to drain the blood from the portal vein and the hepatic artery to the central vein. So blood will go through. So this deals with this blood circulation. So what is the or what is the function it serves by draining the blood from the hepatic artery and portal vein to the central vein? Why blood should be passed from the portal vein to the central vein?
Sir, metabolism.
Yes, because the portal vein, they actually collects the venous blood from the intestine, different parts of the intestine. So these blood will contain the digestive end products, and that should be dispersed to all the hepatocytes for metabolism. So this hepatic lobule serves this function. Now the portal lobule is centered around a portal triad, and on 3 sides there will be 3 central vein, and this is triangular shape, and it is related to the function of transport of bile, because from all this direction bile will be transported towards the bile ductule of the triangle. Okay, so this is the function related to the portal lobule. Another one is hepatic acini. This is diamond shape. On 2 sides it has the triangle and portal triad. Then and on each side, another 2 sides, there are the hepatic acinus. So you can see here the arterial blood will be present mostly along the central zone. Okay, so these hepatic acini, there are 3 zones: zone 1, zone 2, and zone 3. So this is the metabolic lobule or metabolic unit of the liver. So in the zone 1, that is closer to the branches of hepatic artery, so the oxygen content in the zone 1 will be much more Then the zone 2 oxygen content will be less, and in the zone 3 the oxygen content is very few. So the amount of metabolism which will be much more in the zone 1, then zone 2, and then zone 3. Now another two important term that you will face or you will be asked in the written for 2 marks question as well as in the oral part. That is space of DC, and another one is space of
One.
Everyone clear about this too?
Yes, sir.
Yes,
Yes,
sir.
sir.
Space of DC, it is also called perisinusoidal space, right? The space of mole, they are called
Periportal space.
periportal space. So this is the space of DC, that is perisinusoidal. Sinusoid means the gap between the two sheet of hepatocyte. So these are the sinusoid. So perisinusoidal space or the space of DC are the space between the hepatocyte and the hepatic sinusoid. T is a small space that is called the space of DC or perisinusoidal space. Another one is periportal space, that is this. Sorry. So the space of mole, it is the space between the portal canal and the outermost hepatocyte of the hepatic lobule. Now these are the slides you will get in your college. So same slide, two different slides, but both are liver. It is a good one. That is not that much good. It is very lightly stained. So these are the central vein. If you see, so central vein, there will be a small round gap, but if you have to identify the portal tract, so then you will look for three structure together. If you zoom more here, so this is a portal tract, this is a central vein, this is central vein. So if the examiner asked how will you identify this slide as a slide of liver, the answer will be the portal vein and surrounding portal tract and radiating layers of hepatocyte. So these are the hepatocyte. Sorry. From here, these are the hepatocyte. Now, gallbladder. So one important question that is asked or related to the gallbladder is a sac-like structure, you know the function, but why I mentioned the gallbladder here, because there is small difference between the other layers of the intestine and the gallbladder, because you know there are 4 common layers present in the different part of the intestine, but
Yes, sir.
in gallbladder one of the 4 layer is missing. Which one?
Submucous.
Always remember that submucous layer will be absent in the gallbladder. Now pancreas. We know that pancreas is a
Mixed gland.
mixed gland. Why it is called mixed gland?
Because it has
It has
It has
both
both exocrine and
Exocrine
endocrine.
functions.
What is the function of the exocrine part?
Secretion of
Secreting the pancreatic enzyme.
The function of the endocrine part?
insulin.
Insulin glucagon.
Insulin,
Somatostatin.
pancreatic polypeptide.
If you see the histological structure of the pancreas, you will see these are the pancreatic acini or the exocrine part. And among the exocrine part, there are some islands type scattered cell, groups of cell. They are called islets of Langerhans or the pancreatic island. If you see another schematic diagram here, so these are the islets. This is another one, and other part, these are the acinar part of the pancreas. So they are connected to the ductal system, and their secretion will be conveyed through the which duct,
Main pancreatic duct of Wirsung.
and they will be excreted through the hepatopancreatic ampulla or ampulla of Vetter in the second part of duodenum. And the secretion of the islets of Langerhans, they will be secreted and conveyed throughout the body by the blood. Now, one important thing is that there are different types of cell present in the islets of Langerhans. You will have to remember the types of cell, their percentage, and their secretion. So A cell, they will secrete glucagon. B cell, the most important, 60 to 70%, they will secrete insulin. And 5 to 10% cells are D cell, and they are somatostatin. This color code is not that much necessary. The cell type, percentage, and their secretion is important. So this is all about the histology of the digestive tract. Anyone have any question?
No, sir.
Sure.
Yes, sir.
Yes.